So yes, like I was saying, so in these discussions, we'll be going over any doubts or clarifications that any of you might need. And we will be taking them up one by one. And we'll also discuss some sample questions for that week. And once the assignments for each week are already past the due date for submission, we'll be discussing those assignment questions in the upcoming weeks as well. So because this is your first session and your first assignment is still due, we will not be going into the questions for this week, but we will be looking at some similar questions which are brought up from the same topics. And we'll be covering the subsequent assignments in the upcoming weeks as well. So that is the broad idea for this, just a second. Yeah, for this live interaction session. And case any of you have any doubts immediately, I would just urge you to hold on for 5 minutes. I'll give some brief introduction into the topics that were covered, and then we can get to your questions directly, if that is all right. So, are still joining, so we can maybe wait for some time. And also, in case you people do not know, we are recording the meetings. So please do behave accordingly. And that is the only request we have for you right now. And please let me know if the screen is visible. There is a slide that should be currently visible to you. Please let me know if you can see that. You can write in the chat or you can answer directly, whatever is comfortable.
Yes, ma'am. Screen is visible.
Okay, thank you so much. Okay, so let me go over the introduction for this week. So as you already know, the focus of this week was to look at the central dogma of biology and mainly to look at each of the processes. Now, does anyone really know or are any of you already familiar with the difference between cell biology and molecular biology? If anybody is, please, you can answer, unmute yourself and answer. It can be wrong, do not worry about that. Okay, I'll start off by explaining that difference in the beginning. So when we talk about cell biology, it basically means anything and everything that allows a cell to function. So that would include the cell's function as an individual cell. So the processes that take place inside the cell are also a part of cell biology, which is what we consider to be molecular biology. So molecular biology is about dealing with individual molecules. When I say molecules, I mean DNA, RNA, protein, how they individually are synthesized, how they function, how they communicate with each other to allow the cell to ultimately survive. That is what we understand in molecular biology. Now, because these processes, along with n number of other processes altogether allow a cell to survive, they are also included under the purview of cell biology. That is why when we are starting off with the concepts, because this is just an introductory course and we do not go into very detailed explanation for these things immediately. So this course also looks at each of these topics in a more superficial manner. We get an introduction to each of the topics, and then whatever is of interest to each one of you or anybody, you can get into those details at your own time, or you can bring up questions in the meeting. So that is certainly up to you. So that is why the starting point for the course was from the central dogma, which actually is covering the, let's say it gives you a bird's eye view of what happens inside the cell. So let me just open the slides for this week. So in the first lecture, if you remember, Professor Ratnaparki, he had introduced the concept of central dogma, where firstly, the idea of a dogma was discussed, where a dogma is basically a hypothesis where initially scientists, because they did not have concrete proof, or because there are some things you cannot necessarily show within a cell. Like, for example, I think in one of the video lectures, somebody had asked that, can you really see the synthesis of DNA happening within a cell? And the answer to that is very honestly, no, you cannot see it, because it's a process which is occurring at such a low magnitude, which means in the level of angstroms and micrometers, lower than micrometers. It is happening at such a low scale that we do not have the imaging machinery which is capable of monitoring that entire process while it is occurring. We are capable of doing live cell imaging, which means that when multiple cells are interacting with each other, or they're regulating some process, we are able to capture that. But replication is, since it is occurring at the level of DNA, it is much, much smaller, and therefore it is much more difficult to monitor. So that is why, since you cannot always necessarily prove or show these things happening in real time, a lot of these concepts are proposed initially as hypotheses, otherwise known as dogma. So yeah, so that is what the initial discussion was about. It was the central dogma which was proposed by Francis Crick, and it explained how, just admit everybody. Some issue with my internet probably. But yeah, it basically explained how the living cell functions, and how the three major macromolecules within the cell, which are DNA, RNA, and protein, how they are allowing a cell to function at large. So while the more commonly known idea is that DNA gives rise to RNA and RNA gives rise to protein, that is not necessarily the way it proceeds. And instead, there are a lot of reversible or bidirectional interactions that actually take place. So in case of, for example, viral reverse transcriptases, we see that you can see RNA to DNA being formed. And you can also see that there are individually these molecules, since they are capable of replication, they would also produce more and more copies of themselves that would all become a part of the central dogma. So that was the starting point for the discussion. And I think since a lot of people have joined already, maybe you can go ahead and explain or just bring up any doubts if you already had from the session, and then we'll get to the questions later on. And I'll just reshare my screen after. Anybody has any questions they want clarified for now? You can bring them up. Please don't hesitate. Okay, so if not, I'll come to the topics that were discussed. So as we were already discussing, that the central dogma, as was proposed by Francis Crick, it basically showed that DNA to RNA to protein could be synthesized within the cell, but it could never work reversibly, where from protein you would never get RNA, or from protein you would never get DNA. That shows that there is a flow of information that is maintained within the cell, where it is more often than not, it is unidirectional. There is only one way in which information can flow, and that is allowing a cell to perform its function in a defined manner. However, you can still debate one particular concept which is mentioned here, which is the replication of proteins by itself. While Francis Crick in the late 1950s had said that protein by itself will not undergo replication. If you look at the concept of prions, which are they are spelled as prions, prions. They are proteinaceous infectious agents. So what they basically do, they are living proteins. While the term sounds very strange, they are not living in the sense of an organism which is breathing or respiring or which is metabolizing, not in that sense. They are just proteins, but they are capable of causing infections. So there is a particular disease which is commonly known as mad cow disease, which, as you can understand, is it affects cows, and it usually affects their it causes neurodegeneration, and obviously the cow is unable to survive. So such diseases are caused by pathogens, not really pathogens, but since they are causing a disease, they are named as pathogens. So these are not living organisms. They do not have genome. They do not have a RNA or a protein genome which undergoes replication, much like you see even for viruses. Even though viruses are not living, they have a genome which undergoes replication, and that allows them to reproduce. So somebody unmute. So that is basically the only anomaly we find to the concept that was originally proposed by Francis Crick. So later on, there was so yeah, here you see that the central dogma is also known as the central hypothesis. So, like I said, hypothesis is how you initially present an idea and then you use experimental data to back up what you have initially proposed. And your experimental results should justify the idea that was initially brought forward so as to conclusively say that what you have proposed is indeed what is happening inside the cell. And why this is so important is because once any particular scientist, their data gets published or is available to the scientific community, that would be considered as the full and final, or you can say, while that is up for debate, it is something that other scientists will base their experiments on. So, for example, say, if Francis Crick had initially said that, at the top of the dogma sits proteins, and proteins are what are causing, or what proteins are encoding for DNA. Say, for example, it is not the correct scenario. But if he had shown that and he somehow had gotten experiments which backed up this concept, then eventually over the generations, that is a concept that would have had to go into a lot of debate and discussion. And it is why this is why it's so important to make sure that whatever a scientist is publishing, they are publishing it with enough backing or enough experimental evidence to show that indeed the concept that is being proposed is true. So despite that, even when an initial idea is proposed, even 50 years from the time it was proposed, it can still be debated, it can still be questioned, and people get proved right much, much later than the point they were initially proposing. Somebody has a question. Mamta, yes, please. Mamta Saraf, you have a question? Okay, you can interrupt me if you're able to unmute yourself later. You can interrupt me at any time and ask a question. There's no problem in that. Okay. So, like we were talking about the central dogma, so here you see that the initial concept was was rearranged and proposed in this manner, where there was DNA which was giving rise to RNA. Individually, DNA is capable of replicating itself. Same goes for RNA, and then RNA is producing protein. And here you see that what are the machineries that are involved in the process. So the machinery that goes into DNA replication is what we will be taking up towards the fag end of the class today. For the conversion of DNA to RNA, the versions of RNA that are produced can also be different. There are mRNAs, which are messenger RNAs, which eventually get transported. get translated into protein, but there are also tRNAs, rRNAs, long non-coding RNAs, and a variety of other populations. They all have very diverse functions within the cell, and each of them are extremely important for a cell to function in the manner they are supposed to function. So, yeah. So, that was the initial idea. Then we came to the concept of the RNA tie club. So it was a concept that was brought forth by elite scientists from that time who were considered to be very important contributors to this field. And as you can see, it includes Jim Watson and Francis Crick, who were very elite members of this club. And it is something that the scientific community has always dealt with. And additionally, like you can see, there was considering the time that it was, okay, so there might be a little bit of darkness right now. Mm. Yeah. So anyway, just lost light here, so I'll just switch off my video for the time being. So there were some very eminent scientists who were all contributors to this particular club. And as was discussed, they all had been given individual names of amino acids just as part of some unity among the scientific community. So gradually, the central dogma kept on getting redefined in different versions of it. So while the initial version showed that there are all possible flows of information that can potentially occur within a cell. So it shows the bidirectional flow of sequence information. So from DNA to RNA, DNA to protein, protein to DNA, all of this was initially proposed. However, over time, what it kept on getting refined, and it was more clearly defined in the last part of it, as you can see, where under most circumstances, the flow of information is unidirectional, where you get DNA to RNA and then to protein. There are certain circumstances, and only certain circumstances, where RNA can give information, or RNA can be used to derive DNA, whereas the flow of information would be maintained in the same direction. But till now, while it is still quite debatable, till now there hasn't been much evidence that actually shows that protein can actually give rise to information for DNA. And why that is the case is something you will eventually learn once you understand how the process of translation works. So just for now, if we think about it, how translation works is that it usually, when you have a protein, it is encoded by amino acids. So amino acids have a certain property, or rather the genetic code which is followed in the coding of amino acids, that has a certain property which is known as degeneracy. We will come into details about it in the next week. Don't worry about that. I'm just giving you some idea for now in case you've gone through the lectures already. So degeneracy is the property whereby multiple triplet codons. So if you're familiar with codons, they are basically letters, amino acids which are denoted as letters, which when three of them are taken at a time, they encode for one particular like three. Let me rephrase myself. Three ribonucleotides or RNA monomers, when they are arranged in a certain order, they encode for a particular amino acid. So there is some amount of degeneracy that exists in this coding, which is why retracing the sequence or retracing the information which is within a protein back to the DNA is quite difficult, and there can be multiple versions of it, which is why this flow of information is not really supported, or is not really something that scientists or the scientific community believes in. So that is the idea I was trying to get across. Um, then comes the overall concept that is more or less accepted and has been acceptable for an extended period of time till now. So you see that in the process of replication, DNA is duplicating, which is the concept we will be delving into today. Then the next process, or the next major step in molecular biology of a cell, is transcription, which is the synthesis of RNA from DNA. Then comes the process of translation, where we get protein from the synthesized mRNA, and then that protein is capable of performing all sorts of different functions within the cell. So, majorly, while DNA is mainly attributed to be the um, it is the it is the main place where information within a cell is stored. So you would see that you cannot afford, or it is not something that the cell will allow to have mutations or to have any sort of disruption in the information which is contained within the DNA. So because the DNA is the main storehouse of information, and it is responsible for faithfully carrying that information across generations, so that is why it is considered to be the master molecule that holds on to that information and holds on to it correctly. However, when we talk about the functional part of it, DNA by itself is not very capable of performing all sorts of function. Therefore, the functional part is taken over mainly by proteins. And that is why the process of transcription and translation are also as equally important, if not more, as the process of replication. So, this was the first landmark or breakthrough paper that actually brought forth the concept of how DNA might actually be within the cell. Yes, Abhay has a question. Please go ahead.
What is the difference between chromosome and chromatin?
Chromosome and chromatid, right? So, what do you understand by a chromosome? Tell me that first.
Gene carrying substance that responsible to carry the
Um, Sorry, you
parents to
got interrupted. Can you repeat once more? Gene carrying substance.
genes are the substance that allow the transfer of characteristics from the parents to offsprings.
Right, that is a gene. Now, what is a chromosome? What is the difference between these? Are you familiar with it?
No, ma'am, please explain.
Okay, just let me then. Okay, so if you think of a gene, say this is a gene. I'm not sure if it is visible, but right. So think of this as gene X. Okay. Let us take any protein, for example. Say, if you have looked at these lectures, you are familiar with the protein helicase, right? So helicase is, while it is an enzyme, it is also a protein. It's a protein that functions as an enzyme that opens up DNA during the process of replication, right? So this is the gene. I'm just representing it for your understanding, which is coding for, or which is allowing the synthesis of the protein helicase. How is it doing so? The gene is getting transcribed and then translated. Correct? Now, this gene is part of something which is much larger than it. So, I don't really have a cursor here. So, if you think of, okay, it's not working. Say this is, while it's not the best representation, I do not have a cursor on me right now, but basically this gene is part of a chromosome. If we represent a chromosome by X, most of the time you will find that it is represented by X, right? So a chromosome is, how do I make it more understandable for you? This gene will be part of, say, it is expressed, or it is present on chromosome number, say, 16 within the human genome. So the entire, any human cell that you pick up, it has 22 pairs of autosomes and it has one pair of sex chromosomes, right? So this gene is present on, could be present on any one of these 22 autosomes. So those individual chromosomes, they are carrying a lot of information, and they are carrying n number of genes. So the human genome has 30,000 genes, which are encoded in these 22 plus one set of chromosomes. So total 46 chromosomes are carrying 30,000 genes and different parts that are present within them. Now, what your question is that what are chromatids? If you look at the structure of a chromosome, um, let me get a diagram for you. I think that would help much better. Just give me a minute. So, yeah, this should be helpful for you. This is just for representation. So this, is it visible? Just let me know that if you can see.
Yes, ma'am.
Right.
Yes,
So
ma'am.
this is a chromosome, but these individual two strands, these are two chromatids. Chromatids are, they are joined at the center, which you can see as is labeled as centromere. So they are joined at one point or joined to each other by some proteins, some very specific proteins, which make sure that these two are always joined with each other. So that is actually responsible for maintaining the fact that any human cell at any point of time under normal circumstances should be diploid in nature, which means it should have two copies of each gene within the cell. So each chromatid is carrying one copy of a gene and of one allele, and that is being also replicated, or a copy of that is also being maintained in the second copy or in the second chromatids. So the two chromatids have similar information within each other for each gene. That is their purpose. The reason why you need to have two copies is because when one round of replication has been completed and then each cell is dividing into two cells, the two daughter cells should get individual copies of that same gene. So during the process of division, the diploid state, it becomes haploid, or it becomes from 2n to 1n, and those individual chromatids will again undergo replication, will become diploid, and will then get transferred to the daughter cells. Uh, was I able to explain it a little bit? Or do you want me to clarify? I can help you out. I don't have images to help you out. That is the reason why there might be confusion.
Ma'am, if chromosomes are present in DNA, then which among the mRNA and rRNA are responsible for transmitting it from the nucleus to the cytoplasm and process for protein synthesis?
Okay, so where is replication occurring location wise? Within
In
the cell, where does replication occur? Are you familiar with it?
replication of what, ma'am?
DNA replication. Where does it occur within the cell? Is it in the cytoplasm? Is it in the nucleus? Is it outside the cell? Where is replication taking place?
Nucleus, ma'am.
In the cytoplasm.
No. So somebody answered correctly. So all of the DNA that is within the cell, nuclear DNA, it is more commonly known as nuclear DNA, is present within the nucleus and within certain subdivisions of the nucleus as well. I'll not go into details of it, but it occurs within the nucleus. Once that DNA has been replicated, the next step is to go for transcription. We will come to that process in the next class. So transcription is where it will be synthesized into RNA. That is taken care of by a separate machinery. So the machinery that allows replication to proceed is a DNA dependent DNA polymerase. What does it mean? When we say polymerases, they are enzymes which have to take something as a template. What? Somebody is speaking, I don't know. So they have to take something as a template and they have to produce something by taking it as a template. Think of it as a Xerox machine. So what paper you are giving initially is your template, and what is getting Xeroxed is your output, right? So in case of DNA, the DNA that was initially present, the parental DNA is the template, and from that, the daughter DNA, which is being synthesized, is your output. Once that is completed, that daughter DNA is being taken up and that daughter DNA is being synthesized into mRNA. This process is also occurring within the nucleus. Apart from mRNA, I said there are other RNAs which have different functions, which we'll not get into now, but that is also being carried out within the nucleus. After mRNA has been properly synthesized, there are certain proteins which recognize that yes, this is a correctly synthesized mRNA. This has to be converted or has to be translated into proteins. And there are a number of checks which occur for that. We'll read about that in translation of this mRNA into protein. So translate of this has been approved. I think I got disconnected. So I was talking about the process that is occurring within the cell. So after transcription has been carried out, only the mRNA that has been correctly synthesized will get translated from the site, from the nucleus through the nuclear pores. There are pores within the nuclear membrane to the outside, which is towards the cytoplasm of the cell. In the cytoplasm, there are ribosomes, which are very huge macromolecular machineries. They are like large assemblies of proteins. Ribosome is not one protein. There are many, many proteins which come together and which read what is written in the mRNA, and they synthesize a protein using that as a template. So for the ribosome, again taking the Xerox machine analogy, the initial paper that you give, that is the mRNA, and what it produces in response is the protein. So that is how it occurs within the cell. But your question about chromosome and chromatid is completely different. The concept of chromatid is that there are, if you think about these as two, this is one chromatid, this is another chromatid. Together they are joined, and this is one chromosome. There are 23 such pairs of chromosomes within the cell. And all of these together is what forms the nuclear content or the genetic content of an entire cell. And during each round of replication, all of these chromosomes have to undergo replication. If there is any defect in this, it causes diseases. I think you might be familiar with people who have Down syndrome or who have Turner syndrome. These are all very serious and um, very debilitating diseases, which a lot of, they are not very frequent, but you can still see a lot of people around you in real life who suffer from such diseases, and they occur from something as, you might think of it as very insignificant, but something as little as the fact that one chromosome has not been, is not present in that person, or one chromosome has been duplicated in that person. For example, Down syndrome, it is often known as trisomy of 21, which means that chromosome 21 is present in 3 copies instead of 2. So there is an extra copy of one chromosome, and that is causing the person so many defects that their entire lifestyle gets affected because of such a disorder. So that is why the purpose of proper replication and the purpose of proper cell division is extremely crucial to the entire organism. So I hope I was able. Yes, Vikash. Vikash, you had a question? Okay. If not, please let me know if it was still not clear. I'll try to explain it as well. Um, Coming back to the lecture that we were talking about. So we were talking about the structure of DNA that was understood by Francis Crick and James Watson. And certainly you have read a little bit about the controversy that was brought about because of the fact that it was initially, the initial experimental detail was with Rosalind Franklin. Please don't unmute yourself unless you have a question. So yeah, so we were talking about that, and this was the landmark paper that actually showed the molecular structure of DNA and how it forms a double helix, the concept of antiparallel strands, and how that is maintaining that structure, that stable structure of DNA, which is allowing it to be the major storage molecule within the cell. So yeah, so Yeah, so these are the different events that took place within the cell, how it was understood how the two DNA strands are connected, how each of the bases in a base pair are contributing to it. The method that was used to understand the DNA model was X-ray crystallography or fiber crystallography, and where the initial discovery happened was in King's College London, and the major contributor to this was Dr. Rosalind Franklin, who unfortunately was not did not receive the Nobel Prize for this discovery, but she was honored with the discovery of this posthumously, which means that after her death, it was understood that she was a major contributor to this discovery, to one of the biggest discoveries in the history of man, you can say. So yeah, this is a snapshot of that paper. You can go through it. It's written in a quite a readable fashion, so it wouldn't be very difficult for anybody to understand, I believe. So that was mostly what was covered in the first lecture. Uh, in the second lecture, same thing has opened up again, I think. The second lecture, I think, was mainly about looking at the doubts of other people, whatever they had in that context. But we will now move on to the basic structure of DNA. So as we have already talked about it, this was the photo 51, the photo from which, or the snapshot of DNA, X-ray crystallographic snapshot that had allowed Francis and Crick to actually understand how the structure of DNA actually is within the cell. And they had basically understood that there is a the pattern in which DNA is, or the pattern in which the two strands of DNA are orienting with respect to each other is anti-parallel in nature, which is giving rise to this unique diffraction pattern. So while Rosalind Franklin is credited with the process or with the experimental details of this discovery, the analysis part of this result is certainly credited to James Watson and Francis Crick, which is why they are also credited with discovering the process of discovering the structure of DNA. So here you see the similar photo which has been shown, and from there the interpretation was carried out. So this is just a basic idea of what exactly is done. So whenever you want to understand the structure of a protein, there are multiple experimental techniques that can be carried out. But one of the most commonly used techniques is X-ray crystallography. So what is done there, just to give you a brief idea, is that whatever protein you wish to study, XYZ, any protein, that is crystallized. What does crystallization do is that it is freezing, or it is um, capturing that protein in that particular state at that point of time, and is ensuring that within that protein, none of the amino acids, none of the atoms or electrons within each of those atoms of each of those amino acids within the protein can move. So it is frozen in place in that particular state. And then that protein can be is arranged in a certain way within the crystal. Then that crystal is subjected to X-ray, and diffraction of that X-ray by the electrons which are present in that particular protein gives rise to a pattern. Then you study that pattern and you go back and you retrace to the arrangement of electrons within that protein, and you decipher the structure of the protein. So while it sounds simple, it is a very complicated process. It takes several months, and that I am also underestimating, but yes, it takes several months to actually even get good crystals for a particular protein. And once you have good crystals, when we say good crystals, it means that it should have low salt, but it should be uniform, and it has a number of characteristics. It should be able to should be able to withstand the type of X-ray that is coming onto it, should have a uniform pattern, and there are multiple criteria which are to be considered. So it is a very laborious process, but it is one of the most accepted ways of discovering the structure of any protein. Apart from that, there are other techniques as well. There is the currently one of the techniques that is very becoming very popular is cryo-electron microscopy. The reason why it is very much in demand, it is also a very difficult technique. I'm not a structural biologist. I'm not familiar with the details of it. But what I understand is that the concept behind it is to allow that protein to have some amount of movement, because you see within the cell, proteins are not static, they are not frozen in place, which is what X-ray crystallography is doing. It is showing some movement, it is doing some movement within the cell, which is allowing it to exhibit its function. Say, for example, helicase. Helicase is pulling apart DNA so that it can allow the polymerase machinery to move through. So when you take a crystal structure of helicase, it is not replicating those movements, and you are limited by whatever information you are getting from just that snapshot, which is why techniques like these are more desirable, and they are currently very much in practice, and they are opening up new avenues to study many different kinds of proteins. So you can read up on cryo-electron microscopy, at least the basics of it, because it is a very complicated technique, certainly. But yeah, that is another way of studying protein structure. But back then in the 1950s, X-ray crystallography was all that people had. And in fact, now we have a lot of very advanced visualization tools based on which you can actually simulate what kind of a structure the protein might have within the cell. But back then, computer or any sort of technology, computer came much later, but any sort of technology was not available to actually visualize it. So from the proteins, what they actually used to do was make handmade models of each of these. Like if you see here, the structure of DNA that Rosalind Franklin, sorry, that Francis and Crick are standing here with, you're seeing a physical model of the entire DNA. So they used to prepare entire models of whatever structure they were trying to solve. Similarly, goes for every single protein that was understood. So back then, despite all of their limitations, people have made fantastic discoveries, which shows that all you need is a little bit of patience and a lot of analytical capability to get through whatever you want to get through. So next comes the idea that the controversy that we talked about. I'll not get into details of it. You can read up on it. It is one of the biggest controversies in the field of science, and certainly people agree that Rosalind Franklin should have been credited with this discovery as well, because it would have been impossible for anybody to get to the point that they got to without her initial discoveries. So this would be a relevant information for you, certainly, to remember that Francis Crick and James Watson, they received the Nobel Prize in Physiology of Medicine in 1962. So several years after their initial discovery had been made. Now coming to the chemical structure, the chemistry that goes behind the structure of DNA. Certainly, if you want to read this in more detail, you can certainly do so. Please refer to the textbooks that have been provided as reference. You can go for Alberts, Bruce Alberts. You can even go for Leninger. In fact, Leninger has a much better description of it. So biochemistry, principles of biochemistry by Albert Leninger, is these are all very commonly available books. You will get PDFs with this on the internet, and you can just to get an idea of how exactly DNA is within the cell, you would be able to very well understand it. So you can go through Leninger, you can go through Voigt and Voigt. These are all excellent books, and these are the landmark books for at least starting off with biochemistry. But for now, what we can look at is the fact that DNA is made up of two. So DNA, as the name indicates, it is deoxyribonucleic acid. So it is made up of nucleotides, which are forming polynucleotides, and they have, if you think of them as two chains, these chains majorly have a sugar phosphate backbone, and the sugar phosphate backbone is formed by phosphatidyester linkages, and two such strands are coming together and they're twisting around each other to form the structure that we see for DNA. Now, what is allowing them to come together? There are bases, nucleotide bases, which are, they're called as bases because these are ATGC, or adenine, guanine, thymine, and cytosine, are nitrogen bases, which have the structure that you see here. They are either purines or they are pyrimidines, and only only purine and pyrimidines are able to form bonds with each other, and they're able to come together via hydrogen bonds to form the structure where both these strands are attached to each other, and they not attached, they're able to hydrogen bond with each other, and they're able to form the double helical structure that James Watson and Francis Crick had understood and had discovered. Their nature in which they are arranged is usually, the nomenclature for it is done in such a way that they are anti-parallel to each other, which means one sequence runs 5' to 3', the other sequence runs 3' to 5'. So that is the basic idea. So here you see the sugar phosphate backbone I was talking about. So individual nucleotides are connected to each other by these phosphates. And these are the 4 nitrogen bases that we have talked about. And these are purines and pyrimidines, which are together helping in the hydrogen bonding that I showed here. So if you zoom in here, you'll be able to see how A and T are forming double bonds with each other, whereas G and C are forming triple bonds with each other. So because of this nature of hybridization, or this nature of forming hydrogen bonds with each other is so determined or so set in place, therefore the way in which DNA can be arranged is also, it is limited. And finally, from DNA to proteins, what you can see is that the information in DNA gets transcribed into RNA and then translated into protein, like we have already previously discussed. Um, then here you see the basic components of a nucleotide. A nucleotide has the nitrogenous base, the sugar, and a phosphate, which all together form the DNTPs, which can either be DATP, DGTP, DCTP, or DTTP. So these are the ways in which they work. And the difference between the nucleotides for DNA and the nucleotides for RNA are that the deoxyribose, the name DNA has deoxyribose, because in the second carbon, one OH group is lost, and instead there is a hydrogen in its place. Compare that to ribose, which has that OH group or hydroxyl group. This 2' hydroxyl group is the major distinction point between DNA and RNA. And these ribonucleotides altogether, they form the chain in an RNA that we are more familiar with, and we'll get into details about that later as well. So yeah. If anybody has any questions, again, please interrupt me. So yeah. So here you can see that from 5' to 3', each of these different nucleotides are arranged with respect to each other, and every time there is a loss of a proton, and these phosphate groups, 2 phosphate groups are removed, and individual nucleotides are joined to each other to form the polynucleotide chain. So this is another schematic of how the phosphodiester backbone looks like. And then you had a little bit of an example of how DNA is drawn. And this is the phosphodiester linkage in more greater detail, where you can see that this is the phosphate group. And since this is an O linkage, which is formed via oxygen, that is an ester bond, which is being formed with the adjacent nitrogenous bases. The phosphodiester bond is the backbone of the entire DNA molecule. Um, yes, somebody has a doubt. Ajay, please go ahead.
What is the difference between nucleotide and nucleoside?
Nucleotide and a nucleoside, right? So where is that? Yeah. So this is a nucleotide, which is where you see there is a base, plus there is a sugar, plus there is a phosphate. Second, there is some issue. Sorry, there's some issue. Okay, so your question was about nucleotide and nucleoside. So nucleotide are base, sugar, and phosphates. So here, if you look, I think it's a little distorted, but anyway, you'll be able to see here that there is a nitrogenous base. There is the sugar, there's a nitrogenous base. This is your sugar, which in case of DNA is the deoxyribose sugar. And with it, you are able to see this phosphate, triphosphate in particular. However, in nucleoside is where you only have the nitrogenous base and you have the sugar, but you do not have the triphosphate attached to it. So nucleotide triphosphate is NTP. Nucleoside is just the base and the sugar without the phosphate. That is the major difference between the two. Yeah. So after that, we come to the DNA forms. So majorly, there are three forms of DNA that have been understood till date. And initially, these were decoded by Rosalind Franklin during her experiments, where she varied the humidity, salt concentration, and other physiological parameters. And she saw DNA being arranged, the same nucleotide sequence being arranged in different formats. But the most commonly occurring form of DNA, which is what we mostly see in our genomes, is the B form of DNA. Apart from that, there are A forms and Z forms, which also are present in other, which have specific functions within the cell. I'll not get into details of it. You can look it up. It's very interesting. And they have very specific reasons for being present in those areas, like Z form particularly. It's present in very specific regions of the genome for very specific regions. And majorly, the difference between these forms is with the respect of how they are arranged, whether they are right-handed or left-handed, whether they have major minor grooves, what is the repeat, which is the number of bases that are present in each turn. So after how many bases you can see a repeat in the turns. Then the nature in which glycosyl bonds are formed, whether they are anti or syn. I think somebody had asked this in a bonds can mostly be arranged. So if you have, say, two carbons which are bond to each other, and with respect to that, the substituent groups, if they are present on the same side or on the opposite side, that will decide whether the nature of that bond is syn or it is anti. So that is the distinction between the two. If they are on the same side, then that is said to be syn. But the reason why this distinction is made is because when two very large substituent groups are on the same side of the chemical bond or of the covalent bond, what it ends up doing is that it creates a lot of steric hindrance. So those two structures will physically be a little apart. Compared to if they are on opposite sides, there is less steric hindrance, and then the structure of that particular protein will also differ in the long run. So the chemistry behind it is what eventually ends up deciding the structure of it. Then you also see a difference in the helix rise. When we say helix rise, it is that if you have the axis, and with respect to that, you have multiple bases which are arranged one after another, then with respect to the axis from one base to the next, what is the shift or what is the rise in height that you're able to see per base pair? That is also varying from among each of these different forms of DNA as well. Then comes the base tilt, which is that with respect to the first base, how much shift do you see apart from the rise? When we say rise, it is directly one on top of the other. What is the height difference? But from there, what is the tilt also? That can also vary from each of these forms of DNA. And finally, you see a structure of it as well, how the different forms of DNA are arranged. And you can also see these top views. So if you look as your DNA structure, and this is how you're looking at longitudinally. However, when you look at it in this way, I'm not sure if you're seeing it. If you look at it head on, you'll be able to see that there is a central axis, and with respect to that, you'll be able to see that the nitrogen bases are towards the inside of the pipe, whereas all the substituent groups of the sugar phosphate backbone is projecting outwards. So that is how the structure of DNA can be visualized. So here you just have a slide which explains the magnitude in which within the cell different structures can vary. So you have the B form of DNA being present somewhere in the nanometer scale versus you have a lysozyme, which is a relatively smaller protein. If you're not familiar with it, lysozyme is an enzyme which it cleaves the bacterial peptidoglycan, which is the structure present on the outside of the bacteria, and it is present in your tears. All our tears, they have lysozyme, and it is one of the major mechanisms by which it's the body's defense against bacterial infections occurring very commonly in the eye. So most bacteria, there are different types of bacteria as well. There are gram positive and gram negative, not getting into details of it, but majority of the bacteria, they get affected by the lysozyme, majorly gram positive bacteria, which cause a lot of infections. So that is why it's expressed within the tears of the eye. Next, you have the structure of DNA gyrase, and you can see gyrase is response. It is a different type of enzyme which also has a major role to play during replication. It helps in twisting DNA in a certain way so that there is less strain on the DNA. So if you think about it in a way that if a if you're pulling two chains apart, it is creating some strain on the side opposite to where you're pulling, right? So gyrase has the responsibility of relieving that strain within the cell, and it costs a lot of energy, but it has a very important function to do that. It unwinds on the other side so that the strain can be reduced. That is its job. And you also see the structure of ribosome, which, like I said, is a very large protein. It's not just a large protein, it's an assembly of a huge number of proteins as well. And that has the responsibility of synthesizing new proteins within the cell. So that is the job. Here you see the structure of, oh, there are questions. Sorry, please go ahead. I didn't notice. Akansha, Gayatri, whoever, please unmute yourself.
Excuse me, ma'am.
Yeah.
Ma'am, I'm Gayatri here.
Mhm.
Like you thought about the different types of DNAs like bDNA, Z. What causes the changes in their shape, ma'am?
You got interrupted. Sorry. You said what causes?
The changes in the shape, like bDNA is very much different from the zDNA. What is the change it makes it? Why does it happen so?
Why does it happen so? Okay. So if you go to that slide only, I think that will explain it well. Right. So say you have, okay, so let's take your question in this way that your ingredients are the same. You have the same nucleotides which are forming this particular DNA structure, both A and B forms and Z form of DNA. But what is the difference? It happens in how these individual bases are stacked or are arranged with respect to each other. Say, for example, when you have a, say, you have say four nucleotides that you have in your hand, and you are synthesizing DNA. You are responsible for that job. You will, when you place these bases or these nucleotides one on top of the other at a distance of 3.4 angstroms, right? And you are placing it in such a way that in one turn, say from here to here, this is one turn, you are placing exactly 10.5 base pairs of DNA so that you are getting this particular structure. Then your DNA would be arranged in a certain way. In contrast, when you are placing one base on top of the other such that there is a height difference of 2.6 Armstrong, which is much lesser, which means that the bases are closer to each other. But the number of bases that are present in each turn is 11. What is it doing? It is creating a much bigger loop. When you have bigger difference, but a certain number of bases, it is creating one pattern. Compare that to where you're placing the bases close together, but you're accommodating more number of bases. They are more loose with respect to each other, which is why you can see that the width here is much larger in contrast to this. So how these bases get stacked one on top of the other, how many bases are present, what is the way in which they get arranged, all of this will determine how that structure is finally made. And it is not that these are wrong forms of DNA. It is not that. These are present within the cell and they are necessary for certain functions, which is why the cell is synthesizing them in a certain way. What we are focused on is to look at how the genome within the cell, the majority of the DNA within the cell is present, which has the genetic information. That is mostly the B form of DNA, which is what we focus on, which is what we mostly work with as well. So in intracellular mechanisms are ensuring that DNA gets synthesized in a certain way. And the reason behind structural difference is because how they are stacked or how they're arranged one after the other. That is the only difference.
Okay, ma'am. Thank you. I'm clear with this. Do we have the changes in the nucleotides or except guanine and adenine? Like, do we have another type of nucleotides or rather than the four which we study?
There are many different types of variations that you can actually get. Like just from DNA to RNA, you can see, are you familiar with it? RNA has a different nucleotide. Are you familiar with
Yeah,
that?
ma'am, the uracil and the thymine.
Right. So do you know the difference between their structures?
Yes, ma'am, there's a presence of the third methyl group.
Right. So 5-methyl, third methyl group is the difference between the two. So there is, these are the, when we just talk about nucleotides in general, these are the only 5 that we talk about. There is A, T, G, C, and U. Among these, U is a ribonucleotide versus the other four are for DNA. So these are the common variations you will get. Apart from that, within the cell at different stages, each of these nucleotides can undergo certain types of variations. Some modifications are made to them. Like I will not go into much detail about it, but one of the major modifications that occurs is methylation, where a methyl group is attached or a methyl group can be removed. And these little differences among nucleotides, they are responsible for carrying information forward also. Um, the short answer to your question is that these are majorly the 5 types of nucleotides that are within the cell. If I diverge a little bit and give you a different answer to that, if you remember during the lecture, somebody had asked about how, if there are multiple origins of replication within a cell, how do they not end up overlapping? That if there is replication occurring from one side and there's replication occurring from other side, where does it stop? What happens there?
Excuse me, ma'am.
Yeah.
I'm sorry to interrupt. Actually, we are the first year batches. It's not open elective. Our teachers are just given the link to just understand and gain some knowledge.
Okay.
So we don't know the previous lecture. We are the first lecture students getting into this. Ma'am, was I audible?
Okay. I got disconnected. Sorry. Do you have the lectures for the first week, right? Till replication at least.
This is the first lecture, ma'am.
No. So, okay. So this is a live interaction session, but you have the lectures. I think you do. I'm not sure.
I'm sorry.
Okay, if you're not okay, if you're not familiar with it, then I'll hold back and I'll as we get to the eventual slides, we'll come to it. Then just to answer your
Sure,
question,
ma'am.
I'll keep
Sure,
it
ma'am.
short then. There are 5 nucleotides majorly for DNA and for RNA. Sorry,
Sure, ma'am. Thank
Akanksha
you.
has her hand raised for a while. Yeah, welcome. Please go ahead.
Yes, ma'am. My question was, why does the adenine pairs with thymine only? Why does it not pair with cytosine?
Why does it not pair with cytosine? Where is that structure? Just give me a second. So, to give you a short answer, it is because of the type of bonds that are formed. You have to go into the structural details, and you can certainly go for this particular book that I talked about. It is Albert's. You can go to Albert's or you can go into Leninger. These are excellent books for starting off biochemistry of the cell. So you'll be able to see the structures in much greater detail. And if I just give you a short answer, basically, if you see these two bases or these bases among themselves, they form hydrogen bonds, right? So N and H or O and H, they are forming hydrogen bonds with each other. So in adenine and in thymine, the arrangement of these nitrogen atoms and the hydrogen atoms are such that they are able to form 2 bonds with each other. If your, for example, your question was, why does adenine not pair with cytosine? Cytosine has 3 available sites through which hydrogen bonding can actually occur. If it pairs with adenine, it will have free hydrogen left behind, which will not be able to pair with free nitrogen left behind, which will not be able to pair, and that would make it less stable. The structure of each of these individual nucleotides or each of these individual nitrogenous bases is such that they are perfectly complementary to each other. So adenine has 1 nitrogen, and one hydrogen, which are able to hydrogen bond with the two available sites present on thymine. And also G and C are able to form similar three hydrogen bonds with each other. Technically, they could form multiple interactions. A could bond with C, T could bond with G, but they would always be less stable structures. And what DNA always tries to do, which is the reason why DNA is the master molecule that carries all of the genetic information within the cell, it is because it forms the most stable structure which is possible, which means it has the lowest free energy that it could under those circumstances. So that is why the bonding is arranged in such a way that it optimizes this bonding among the different nucleotides. I hope that answers your question. Sorry, I keep getting disconnected. Ajay, you had something to ask? I think you had raised your hand. Okay, if not, we will proceed forward. So we were at the different forms of DNA, and the size scale was talked about. And finally, this is about the organization of DNA within a cell, which is where DNA is associated with several types of DNA binding proteins. So okay, so this actually would also somewhat give an idea about a previous question asked by one of the students. So where they had asked about chromosome, chromatids, and a gene. So if you just look at this particular, this particular diagram, it is, consider that you have a chromosome in hand and you are unwinding that chromosome. You are opening up that chromosome. What you will eventually come to is the double helix, the DNA double helix. But within the cell, which has such immense amount of DNA, like I'm not sure if I'm saying the numbers right, but supposedly within each cell, it has approximately, I think, 5 meters of DNA. And you imagine the size of a cell, which is in microns. How will it accommodate so much DNA? It is able to do so because DNA within the cell is packaged and is like it is compacted and packaged into such small okay, if not, we'll move on. So so yeah, so this is how the packaging within a cell, or packaging of DNA within a cell, is actually carried out. So here you see there are a lot of proteins which are present around which DNA is wound around, right? So these proteins, these DNA binding proteins, are called as histones. So these histones, which you can see as H3, H4, H2A, H2B, let us think of it as this. If you have the complex of histones, or the histone octamer present like this. I'm not sure. Yeah. So this is a mouse basically. You have a complex of proteins which are present. Around this, you are winding your DNA. What is it doing? It is allowing the accommodation of a very large amount of DNA. So all that wire is getting accommodated into a small space. Imagine this being done for meters and meters of DNA. It will be able to compact everything and accommodate everything into a very small space. So this is why such a small amount of DNA, or such a small volume, or such a small space is taken up by such huge amounts of DNA within the cell. And what it is eventually done is, using such proteins, you are compacting or condensing genetic information within the cell into chromosomes, and those chromosomes are then carrying it forward over and over generations. So that is the idea behind compacting, and this is carried out by histone proteins, as you can see here. And these histone proteins are forming the beads on string structure, which I just showed using that mouse as well, where DNA is wound around each of these nucleosomes. Then they are further compacted or further packaged by some other proteins to form fiber-like structures. Those fibers eventually form the chromosome. So that is allowing the entire genetic content to be maintained within the cell. So that is basically the idea of how DNA is packaged. Then comes we'll come to the next lecture, and here also majorly the doubt questions were discovered. Just go over the lectures once. Yeah, it's the same. Same thing has been included everywhere. It's not there. Yeah, it's here. So now we'll come to each of the processes now. So till now, what we have gone over is the central dogma, the structure of DNA, the chemical structure of DNA, the different types of DNA that are present, how it is present within the cell. Now that is the basic idea and where it is present within the cell. Now the next part that we come to is the processes that it is actually involved in. Now we come to the details of the dogma, that what is, we have the DNA, what do we do with it now? Yeah, Abhay, please go ahead.
In the previous picture, it was written chromatin fiber. Is chromatid and chromatin fiber the same?
So chromatin, when we say, okay, so there are a lot of terms that are interchangeably used. In the picture that you saw, chromatin fiber was for one particular stage during packaging. So you start off with completely opened DNA. From there, you twisted around nucleosomes, and what you twisted around histone proteins, and the structure that is obtained is a nucleosome. These nucleosomes are then arranged around us. There are some other proteins. So how do I explain it? So you saw this analogy till here. It is a nucleosome, okay, where the DNA is getting packaged around histone. These structures are getting packaged around, say, something like this, a cylindrical structure. They are packaged around such a structure, and they form like a cylinder, a larger cylinder is formed, and that bigger structure with a lot of DNA inside it is forming the chromatin fiber. That fiber is getting condensed further and further, and eventually that is resulting in the structure which you are able to see inside the cell under a microscope as chromosomes. That is what you see finally. Chromatid is one of the two chromosome, like the two sister chromatids are forming one chromosome. That is a chromatid, that is clearly defined. Chromatin fiber is one of the stages of packaging. But there is also another term which is chromatin. Chromatin is which is like during I'm not sure if you're familiar with it, but during cell division, what happens is every time a cell undergoes division, the entire DNA within the cell, which you have put so much energy, so much proteins in, and you have condensed it to form chromatids, or you have condensed it to form chromosomes eventually, all of that has to be opened up. That has to be opened up. Why? Because replication has to take place. A cell needs to duplicate whatever DNA it has so that a copy of the DNA can be transferred to each of the daughter cells, each of the new cells. So during this process, this unwounded or opened DNA or opened chromatid chromosome is called as chromatin as well. So this unwinded version is also chromatin, and a middle stage or an intermediate stage of packaging is also known as chromatin fiber, but it is completely different from chromatid. Chromatid are one of the two. So these are the two systems. Remember this image, like there are two chromatids. They get separated during cell division. They are copies. They each carry copies of each gene or each allele within them. And together they form a chromosome. This is all you can know for now. I hope that is a little bit clear.
Yes, ma'am.
Okay. So here we were talking about the processes that we now get into. So replication, transcription, and translation. We'll take up transcription and translation in the next class. For now, we just go into replication. So what is the protein that is carrying out replication? It is the DNA-dependent DNA polymerase, as you can see here. There are also many other types of polymerases. Like I explained, that polymerase simply means something which will take monomers and will form a polymer. That's all it will do. So for that, it needs something as a template, and it needs to produce something as a result or as an output of it. And for that, you need to have monomers available for it. So when it can either be DNA dependent DNA polymerase, where DNA is the template and DNA is the product, DNA dependent RNA polymerase, where DNA is the template, but RNA is the product, which means this is the enzyme that operates during transcription. It takes the DNA sequence and it produces an RNA in its response. Then RNA-dependent DNA polymerase uses RNA as the template, which is something you see in a large number of enzymes, viruses, retroviral viruses. They usually contain these enzymes for reading RNA but producing DNA in response to it. There are RNA-dependent RNA polymerases also. Relatively less occurrence of these enzymes can be seen, but they are also equally functional. So they read RNA and they produce RNA. And there are also RNA-dependent amino acid ligases, which is what the ribosome basically does, that it reads RNA and it synthesizes polypeptides or it synthesizes proteins. So these are the different enzymes that are operational in different stages of the molecular biology processes that occur within a cell. So that's what we see here. And after that, okay, so this had an schematic representation of how you can draw DNA, and that would be up to everybody's own. People are still entering. Okay, own habits. So for your practice, please refer to those reference books if you are trying to understand the entire structure of DNA. Look at the animations that were suggested in these lectures, and you can also go for the textbooks for certainly. And for your own practice, try to draw out individual um, at least try to draw out one nucleotide and see how from a base sugar and phosphate you are getting a polynucleotide structure. That is very important. Then comes the semi-conservative nature of DNA replication. So it would be understood by this schematic here as well. Splitting, I think. No. Okay. So here you see that this is what you understand or what is usually represented as a replication fork. Why fork? Because it is bifurcating like this. There are two different strands of DNA from the original parental DNA. So what happens here is that DNA is getting synthesized. DNA is getting synthesized along each of these two strands. So the two separated strands of DNA are individually taken as templates. And in response to those templates, new strands of DNA are being synthesized. Why is it called replication then? It is because so you have two DNA strands. One is say A, T, G, and C. Then what would its complementary strand be? It would be T, A, C, and G, right? So these two strands are getting separated. When you are using ATGC, the first strand, as your template, you are synthesizing TACG as your daughter strand and vice versa for the other strand. What happens is from the initial two strands, you now have a complex of four strands. These can get divided between the two cells that will now be formed from the parental cell, and therefore your entire process of DNA replication is getting completed in this manner. So that is why the process of DNA replication, it proceeds via a fork where you have two different strands that are getting separated, and the enzyme, the enzyme that I've been talking about the entire class is helicase, the main protein that is responsible for the separation. Helicase, how do you remember the name? You think of it as the any enzyme that has A's in its name, right? It does some sort of breakdown. It breaks something up, or it separates something, or it cleaves something. That is its job. What it is doing is it is opening the helix, hence the name helicase. So the DNA helix, it attaches to some place, it opens up the DNA and it creates a strain in the other direction. Like you can see here, it is creating a strain in the part that is beyond the point where replication is occurring. The points at which replication start are usually known as the origins of replication. So origin is decided, there is no particular way to really know that this is the origin. What you basically look at is the sequence. So there are some conserved sequences that are maintained within the genome, which dictate that yes, this is the point from which origin or replication would begin. When it is a simpler genome, say for example for a bacteria which has a relatively smaller amount of DNA to be duplicated, it usually only has one origin of replication. So from one origin, two sites replication would proceed and it would complete the replication of the entire DNA. On the other hand, when you have a very complex genome, such as in case of humans or in case of eukaryotes in general, where there is a large amount of DNA to be duplicated, in under such circumstances, multiple origins of replication are present. And they can be in the like in the entire genome, they can be in the number of thousands as well. And this is necessary because for such a large amount of DNA to be duplicated in very limited time, you need replication to start from multiple places or to fire from multiple places at the same time. And then all of the DNA should be faithfully, correctly duplicated without mistakes or with as few mistakes as possible. So that is the idea behind carrying out replication from these places. Now, once helicase has attached, what it does is it opens up DNA in a certain region. So you had very tightly wound around, you had bases that were hydrogen bonded to each other, and that was forming a tight DNA structure. This enzyme goes in between, separates these bases, and what it does is it opens up DNA so that all of the different proteins that are required to allow DNA replication to proceed, they would be able to um, they would be able to dock on that surface and they'd be able to perform their designated functions. So for that, this replication bubble is formed, and from each of these bubbles, replication can proceed. That is the whole idea behind it. So what it effectively ends up doing is that because after helicase has opened up, two separate polymerases, DNA polymerases, are able to bind to each of these different strands. And what they do is they start DNA synthesis. Since the orientation in which this enzyme can move, or the orientation in which the Xerox machine can copy, let's think of it that way, it's fixed. It can only do its function in 5' to 3' direction. But the strands are anti-parallel to each other, so only one of the two strands can allow the enzyme to move in this particular way, which is what we call as the leading strand. So for the leading strand, helicase is able to synthesize or is able to polymerize continuously. It is able to read the template and is able to synthesize the new strand at the same time. But for the other strand, which is in the opposite orientation or is in the anti-parallel orientation, polymerase is not able to do this. And there what it does is it carries out discontinuous synthesis. A few bases are opened up, polymerase synthesizes, again it is waiting, again a few bases are opened up, again polymerase synthesizes. So it does discontinuous synthesis of small fragments, each of which are called as Okazaki fragments. These Okazaki fragments are then attached to each other or are ligated to each other to form one continuous polynucleotide or DNA sequence at the end of it. Yes, Akanksha, please go ahead.
What do you mean by plasmid and what does their role play in bacteria?
Once again, sorry, what do I mean by?
What do you mean by plasmids and what does role they play in bacteria?
Okay. So plasmids are okay, you're not familiar with the term. They are circular genomes. So if you think of a circle, a DNA that is completely closed, mostly we see DNA as like a chain, right? And then that chain is wound into chromosomes. Compared to that, bacterial genomes, under most circumstances, they are circular genomes, which are covalently closed. They are a complete, they are like this. They are a completely closed genome. Those are mostly known as plasmids. Now, apart from the bacteria's own genome, it can a number of times, it is capable of having some extra genetic information within it as well. If I give you an example, you might have heard about the fact that nowadays a lot of doctors, they say that you should not discontinue any antibiotic treatment abruptly. If a doctor has asked you to take medicines for 7 days, you should take it. The reason this is prescribed very strongly is because when you don't do this, when you abruptly stop taking medicines, what happens is that over time there is something called as antibiotic resistance that comes about. Where does this resistance come from? This resistance comes from antibiotic resistance plasmids. Plasmids are also genetic material. They are also DNA. They are covalently closed circular DNA. And that DNA has some sequence which encodes for some sort of protein, that allows that antibiotic to be degraded. Say you give any medicine, any common medicine that is taken, say streptomycin, or you might have heard of penicillin, or there are many antibiotics that are given. So a lot of bacteria, they are over time, with the excessive use of antibiotics among human beings, what happens is that they are acquiring such plasmids or such DNA, which is allowing them to survive against that particular antibiotic, and they are able to be resistant to it. So plasmids are one such way in which they are able to acquire such resistance, and they are mostly carried among bacteria. Only some other organisms also have plasmids, but mainly we see them with respect to bacteria. But apart from that, the bacterial own genome, their own cellular genome or bacterial genome, is also circular in nature. And I said that with respect to such circular genomes, there is only one origin of replication, or one sequence from which replication will start, and then it proceeds bidirectionally in both directions, and the entire genome is replicated. That was the intention of what I had said.
It means that the plasmids help out DNA replication.
Now, your voice is not clear. What did you say?
As far as you said, that plasmid is helping out the DNA replication, which is true.
Plasmid is helping out DNA replication.
Yeah, means plasmid is the basic role of that.
Can you type the question in the chat box, please? I'm not able to understand you. Plasmid does not help out replication. Replication is carried out by a machinery, a replicative machinery, which includes helicase, which includes the polymerase, which includes gyrase as well in some stages. It includes ligase, it has alpha primase. There are multiple, there is a machinery which assembles. When a cell decides that now it is the time for that cell to replicate its DNA, there is a lot of signaling that goes on inside the cell, which decides that now is the time that a cell will duplicate. You know what happens when a cell doesn't duplicate properly, or when its DNA doesn't duplicate properly? It leads to cancer. As simple as that. It is that crucial a process that when you don't duplicate cells properly, when you don't duplicate the genome properly, it leads to very severe diseases. So it is carried out by a replicative machinery, not by plasmids. Plasmids I gave as an example of another type of genome where there is where, because it is circular in nature, there is only one origin sequence. It has nothing to do with the process of replication. Yes, Pulak Das, please go ahead.
Ma'am, why DNA polymerase is working in different direction in lagging strand?
So in which direction does normally polymerase work? Do you know that?
I mean, leading strand it is working normally. So why it is not repeated in the lagging strand also?
Because its movement is oriented in one direction. So because you have two strands, think of them as two strands like this. Okay. In DNA, one of these strands is going from 5' to 3'. If you look at the figure here, that should help you. Say, since this is your leading strand, right? And what is this red arrow indicating? It is indicating the direction in which new synthesis is happening. Okay. So this is your, for the leading strand, the lower strand, which is your leading strand. For this strand, this end is 5' and this end is 3'. Okay. With respect to that, sorry, I am extremely sorry, I'm wrong. This end is 3' and this end is 5'. Once it has been opened up, synthesis is being carried out in this direction. With using that as the template, synthesis is being carried out in this direction. For the lagging strand, which has the completely opposite orientation, the polymerase will not be able to move because it can only read template in one direction. These enzymes, how do I explain? If you looked at the structure of gyrase in one of the previous slides, you could see that there are, within the structure of the protein, it is limited in such a way that movement is only allowed in a certain direction. So You can debate why aren't there separate polymerases for separate strands so that it could continue simply in one way. That is a debatable question. And I think that if it was really that important a necessity for the cell, over the course of evolution, we would have had separate polymerases for separate strands and they would be able to continuously synthesize. But one of the reasons, and I'm guessing this is could be one of the reasons, is because it would be energetically very expensive for the cell. Because any protein that you synthesize, any protein that a cell synthesizes costs a lot of energy and a lot of resources for the cell. So if you expend so much energy into just one process, which is replication, even though the cell already invests a lot of energy into it, if you make it more expensive by needing separate proteins for executing the same function, that would be more expensive for it. On the other hand, if it is using the same enzyme for the different strands, but is just using another enzyme which is ligase that is anyway present in the cell for other functions, it is just recruiting ligase to the site where the Okazaki fragments have to be joined. It is more energetically feasible for the cell and it is able to execute the same function without unnecessarily spending energy. I think that would be the reason why separate polymerases are not required, and why the same polymerase, despite its limitation, is carrying out this activity over time and over generations. This has been maintained. So that is my guess or my idea behind why the cell functions in this way.
Okay, thank you, ma'am.
So this is the X-ray structure of DNA polymerase 3 of E. coli. So it has several, it is actually considered analogous to a palm, the structure of DNA. Again, you can go back to Leninger for a better explanation of this. They have basically thought of it as if this is an enzyme. So if you just read here, these are your finger domains. This is the palm, the thumb, and the DNA is passing in between this, and it is allowing the DNA sequence to be opened up, to be replicated, and the new DNA to be synthesized. So another example of where X-ray structure is extremely crucial. But at the same time, any movement that might be occurring in this particular protein would not be recognized or would not be identified using X-ray crystallography. And for that, you need the more advanced techniques such as cryo-EM. Even NMR is capable of, NMR is nuclear magnetic resonance. It is capable of capturing some movement in proteins, but it has other limitations where the size of the protein is limited. It has to be a soluble protein. So there are other limitations to that technique. So cryo-EM is the more up and about technique that is being explored in the in the current times. So that is what we see here, and we see how the enzyme, so you can see this analogous to a hand, where you can see that the thumb domain is holding on to the RNA primer that is starting off a replication from that process. And this is the RNA primer that is sitting here, the exonuclease domain that will get rid of the RNA primer, the palm or the catalytic domain, which will actually carry out the polymerase activity, the finger domain that is holding on to the ssDNA, which is single-stranded DNA. So what it essentially does is it maintains, or it makes sure that after helicase has separated the two strands of DNA, they should remain separate, because helicase is spending a lot of ATP, a lot of energy within the cell to actually maintain that there is the separation of these strands, and that is being further maintained by the enzyme itself. And using the polymerase or the catalytic domain, it is adding new nucleotides with respect to the template. So this is the part of this lecture. I skipped a slide here, probably. So probably it has the same information. Yeah. So these were basically the slides that were part of the first week. I'll now go through some of the sample questions for this week. So basically, your assignments have similar questions, not the same, but similar questions. So this would give you some idea of how you could actually tackle these. So if you see the first question, it is asking that in the following figure, which pathway for the flow of information has not yet been discovered in eukaryotic cells? So this is the first version of the central dogma that we all saw that Francis Crick had elaborated upon. Somebody's waiting. So this was the first version of the central dogma, and there you can see that all the possible combinations of information flow were indicated. But obviously, over time, we now know that that is not actually the case. So among these, what are the different options? So okay, so whenever you get, not just limited to this assignment, but whenever you have multiple choice questions, what I would suggest should be the way to tackle questions, anything that you are not absolutely sure about. If you're sure about some question, you directly go ahead and pick the right answer for it. But if you are not, always try and read the questions at least 2 to 3 times so that you understand what it is asking. Because when you go for multiple choice questions, especially in competitive exams, if in the upcoming years you go to look, you go ahead and appear for something like GATE or CSIR, DBT, etc., exams like these, there they will present the question in a way which can very much confuse you. So always make sure you know what the question is asking for, then start looking at the options. Now, the options more often than not will have some, like if there are 4 options present, they will have at least 1 or 2 that you can immediately remove. So always go by the process of elimination and you remove whatever is obviously not the answer. And then you try to get your knowledge, whatever you had in mind, and try to arrive at a conclusion. This you should always opt for questions that you're not absolutely sure about, and don't try to leave questions, try not to leave questions unanswered as much as you can, because more often than not, if you've studied something, you will be able to derive some answer out of it. So coming back to the example questions, here you can see the different options that are given. We'll go by the options only. Our process 7, 2, 6, and 4. So 7 is for DNA to RNA. 2 is for RNA replicating by itself. 6 is for DNA to protein, and 4 is for RNA to DNA. So if you read the question correctly, it has asked that which has not been discovered. So obviously 7, which is the process of transcription, has been discovered. Second is for RNA replication by itself. It has also been discovered. And RNA to DNA, which is indicated by 4, has also been studied in viruses, retroviruses particularly. So the correct answer to this would be question 6. Sorry, the process which is indicated by number 6, which is from DNA to protein. So till date, there has not been any example which has shown that directly from DNA you can get protein. It has never been done. There is always an intermediate that is involved, but you never know. Someday in the future, we might get to know some sort of research work which disproves this, but till date, that has not been studied. Then the second question is asking about the first accurate model of DNA and who it was proposed by. So under options, we have Maurice Wilkins and Rosalind Franklin, James Watson and Francis Crick, Linus Pauling and Robert Corey, or James Watson and Rosalind Franklin. So while the X-ray, like we have discussed this, while the X-ray crystallographic structure was brought forth by Maurice Wilkins and Rosalind Franklin, they are still not credited with the discovery of the model for the same, and that credit certainly goes to James Watson and Francis Crick. So it's actually, even if you would go through some of these textbooks, you would see that the base pairing of DNA that is actually known as Watson-Crick base pairing. They are credited with identification of that as well. Yeah, Ajay, please go ahead. Ajay, you've raised your hand. Please go ahead with your question. Okay, if not, please interrupt me later. So yeah. Waiting. I think I got disconnected. Ajay had a question, I think. If not, I'll come back to this. So yeah. The third question asked about, does DNA-dependent DNA polymerase make any mistakes while replicating DNA? So you would expect the second option to be correct, that the replicating machinery does not make any mistakes, and over time, over the course of evolution.
It is
Yeah.
not mentioned in option.
Yeah. What is your question? I couldn't hear. Ajay, can you repeat? I couldn't hear this question. You can type in the chat box if you want. I couldn't hear you. Yeah. So we were talking about the accuracy of the
Can
enzyme
protein
polymerase.
form DNA? Option number 8.
Can protein form DNA? No, it cannot.
First
It
question.
was. It cannot. If you remember the first slide about what Francis Crick had proposed, even in the 1950s, that is what it was. He had initially said that this was the flow of information, and information can flow in this direction, but never from protein to DNA. I explained about this in the beginning as well, that degeneracy, you can speculate what could be the sequence, but you'll actually understand this once we cover translation in more detail. You'll be able to see why this seems like such an impossible scenario. I wouldn't say impossible, but it would give you so many possible combinations that it could be any of those combinations. That was the original sequence. So from protein, you cannot go back to
No,
DNA.
madam, there is word of prion. Prion is there. Prion. Prion is there. Also, we
Prions, yeah.
have scrappy and Kuru diseases.
Yes, I
In that,
mentioned that. Prions are
there
different.
is a
Prions,
the
yes.
protein can form
Prions
DNA
do not encode for DNA. Prions are proteins by
and
itself which
DNA
are
can form
capable of
protein.
causing
That's why.
diseases. Yes,
What
please
about
go ahead.
Kuru and scrappy?
Scrapie is also a prion only. It causes mad cow disease, PRPSC. I'm not familiar with Kuru, that disease I'm not sure about, but yeah, prions are, I guess it is also a prion disease only. But yeah, they are protein, they are just proteins by itself. We still till date do not know how they are able to do that. But okay, we still don't know how they're able to do that. But basically, what they do is they are causing the disease without going via the DNA. They are just
Unless protein can form DNA, this disease
the
cannot be
proteins by
transmitted.
themselves
Means for
causing
transmission, you
disease.
require DNA. Na?
No, they
For
are just
transmission,
proteins.
you
To
require DNA. Unless the protein they are
just
forming DNA.
please let me finish. Just a second. Yeah. So that's what I'm saying. To the best of my knowledge, I can still check it again if you want. But for
Even
to the
in
best of my knowledge,
also that CJ syndrome is said to be, if person is suffering from CJ syndrome, its organ could not be transplanted. Means that is a severe, not all over
let
the
me
world, in even organ
convert it back into
drive
protein.
also. That's why there is that question.
I could not understand you. Sorry. What organ? What?
In organ
I think somebody muted you. Whoever is doing this, please don't. You would be removed from the meeting if you do it. I could not hear your question, Ajay. Please let me know in the chat box later. But yeah, I'll check what you're suggesting. But as far as I know, proteins do not get translated or untranslated into DNA. Proteins themselves are producing proteins. So that is the extent of my knowledge of it. I might be wrong. I'll check it and get back to you with the next session.
transplantation is there. Anything can be accepted except that CJ syndrome, which is just like Kuru and Scrappy, because there is a threat it may be transmitted from one person to another person. And if something is transmitted that is not via protein,
Why can it not be transmitted via protein? Why do you think so? Could it not be RNA? It could be RNA as well, right?
via DNA.
What? You're not audible to me. See, if you want me to specifically go into these diseases, I can go and check them and get back to you. But your voice is getting very frequently disconnected. I'm sorry. I cannot hear you properly. But yes, like I said, I'll get back to you after I check it because I'll not speak about something I'm not familiar with. So I will look into these diseases. But last I read, I knew that prions are yet to be decoded completely, how they exactly work. And prions do not necessarily get transmitted, at least with respect to scrapie, but I don't know about the other two diseases, so I can look it up and I can get back to you in the next class. And please allow me to move forward because I have to wrap up today's session. So coming back to question 3. So here we were talking about the fidelity of the process of replication,
Okay, thank you.
which is which is about the error rate at which DNA-dependent DNA polymerase works. So it has approximately one mistake for every 1,000 to 10,000 nucleotides. So despite all of the different mechanisms that are present, which ensure that DNA replication proceeds with as much accuracy and fidelity as possible, despite all of that, still some amount of mistakes are incorporated. And at the same time, there are many mechanisms that operate and that are in place to make sure that these mistakes are still quite minimized. And one of those mechanisms is MMR, which is mismatch repair mechanism. There are others also in place, but MMR is one of the ways in which a cell makes sure that the number of mistakes is as minimal as possible, despite what mistakes polymerase might already be making. Then comes Chargaff's rule, which is for DNA, which states that if you go through the options, there are multiple choices that the number of purines to pyrimidines are actually twice, which is not true. A and T form double bonds, G and C form triple bonds. That is also not Chargaff's rules. That is just the way in which these bases they pair with each other. But instead, what Chargaff's rules state is that purines to pyrimidines is always maintained in a 1:1 ratio in DNA. And the reason for that is obviously quite understandably because each purine has to pair with a pyrimidine. So there would never be an unpaired base within DNA. They would always be complementary to each other. And as a result, the number of purines that you see on one strand would be equal to the number of pyrimidines that you see on the other strand. And therefore, the ratio is always maintained as 1:1. Then, the fourth option is obviously not correct, which is that there's no relation between purines and pyrimidines. So that is not true. Instead, the ratio is 1:1, which is maintained within the cell. Then in question 5, what did the X-ray diffraction pattern of DNA produced by Rosalind Franklin suggest to Watson and Crick? So that DNA is made up of purines and pyrimidines. It has a double helix structure, it has a phosphate structure, or it has a triple helix structure. So all of the other information that you see here could not be decoded directly from the diffraction pattern. What could be understood from there was that there were two helices, which is why you got that X pattern of diffraction, because these two helices, if you look at the cross section, it comes out as an X. So that is what was interpreted from the diffraction pattern produced by Rosalind Franklin. Then in question 6, we are being asked which of the following statements about DNA replication is not true. So A, still joining. Yeah, so A is that Okazaki fragments are found on the lagging strand. DNA polymerase replicates DNA from 5' to 3' direction only. DNA helicase unwinds and separates the two strands of DNA, or that it is replication is continuous on both leading and lagging strands. So obviously the last statement is incorrect. We say that there is one leading strand where replication is continuous, whereas on the other lagging strand, it is discontinuous in nature, and instead it produces Okazaki fragments which have to be attached or which have to be ligated to form the continuous strand. So again, by the process of elimination, you can narrow down to the correct option. Coming to question 7, we have that two strands of DNA are joined by A, phosphodiester bonds, B, covalent bonds, C, hydrogen bonds, or D, none of the above. So this is something that you actually need to be a little bit careful about when you answer, because all of these different bonds are present within DNA. So the phosphodiester bonds are, of course, in the backbone. Covalent bonds are between the sugar, the nitrogenous base, and the phosphate that is present. But hydrogen bonds are actually what are allowing the two strands of DNA to find that complementarity among each other and to hybridize with each other and to ultimately form the double helical structure that we talk about. So that is in question 7. In question 8, we're being asked that which enzyme joins the Okazaki fragments on the lagging strand, and the options are DNA-dependent DNA polymerase, helicase, primase, or DNA ligase. And the answer to that would be ligase only because it is responsible for the ligation process. Primase, on the other hand, adds the RNA primer, which is required initially for starting off the process of replication. Helicase is for opening up the double-stranded DNA helix, and polymerase is for incorporating new nucleotides to form the polynucleotide chain. In question 9, we are asked that if a single molecule of DNA, which is a double helix, undergoes 5 rounds of DNA replication, 5 rounds, how many molecules of DNA are created which do not have any original strand of DNA? So this is something that I would, if you have already not noted it, you can try it out by yourself. Basically, what you need to make sure is that you are starting off with 2 parental DNA strands. And what you can do is you can use different colored pens or pencils, whatever is convenient for you, and you can mark those 2 strands. Now, from those 2, you are synthesizing daughter strands, and it is undergoing 5 rounds of replication. So you start with 2. In your first round, you have 4. In your second round, you have 8, and you keep on doing this. And from start to finish, what is the total number of strands? From there, you just subtract the parental strands, and you'll narrow down to 30 strands of DNA, which are being synthesized. So you can get similar questions in your assignment as well. And finally, the last question is about nucleotides, which are composed of phosphate, sugar, as well as base. I'm not going through the options because we have just discussed that this is the actual way in which nucleotides are formed. There's a question from Ajay. Please proceed.
Hello, madam. Can you hear me?
Yeah, go ahead. I can't hear you
Actually,
now.
backbone, but I heard the phosphate they are phosphate
Sorry.
they form the backbone of the DNA.
Mm-hmm.
But actually, the 2 DNA strands they are attached with the hydrogen bonds in between. So backbone
Mm-hmm.
is something which is in the middle, right? Giving a rigid
No.
structure.
No. Which bond do you think is stronger, a hydrogen bond or the phosphodiester covalent bond?
I think hydrogen bond needs more energy, especially C and G, to break down rather than a phosphate bond. So hydrogen bond should form a backbone of a D and rather than a phosphodiester bond.
I think you need to revise maybe a little bit of chemistry regarding this, but no, hydrogen bonds are not the stronger bonds. You can think about it in this way. The backbone of DNA is what is rigid. Your concept regarding that is fine. But the rigid part is the phosphodiester backbone. It is the phosphate and the individual sugars that are getting linked to it, which are forming that rigid backbone. What is continuously, every generation, what is continuously getting separated, dislodged, non-separated, all of that is happening to the bases that are in between. If you think of it as a zip, right? You think of a zip, and the sides of the zip are the 2 backbones of 2 individual DNA strands. When you unzip it, they are getting separated and they are forming new strands in between. Again, you zip it up and the replication fork is closed. So what is in the middle are the hydrogen bonds. They are much weaker in comparison. And that is necessary because you need to allow them to dissociate. You need them to get separated every time DNA needs to undergo replication. It is a necessity for those bonds to be weaker than the backbone bonds. And the reason that backbone is so strong is why you need very specific enzymes to break it. You cannot just break DNA just by doing anything to it. You need to have specific endonucleases or exonucleases, which are as the name indicates, they are nucleases. They cleave nucleic acid. So they cleave those backbone bonds and then they are able to break DNA. Or you need radiation, or you need some sort of external agent to break, you need some sort of powerful agent to actually break that DNA backbone. Otherwise, it doesn't break. And when it does break, it causes a number of problems, which is one of the reasons why you're able to kill cancer cells by radiation, because it randomly introduces breaks in the DNA in you hopefully what you hope are cancer cells, and then those cells die because they're not able to repair all of that damage that is within those cells. So those bonds are much stronger compared to what is in the center. But on the other hand, when these hydrogen bonds that are between these bases, when they stack among each other, they provide excellent amount of stability to DNA. So if you are ever asked which bond is giving the maximum stability to DNA, that is always hydrogen bonds, because these individual backbone bonds are providing strength to individual strands of DNA. But when it is forming the double helical structure that we have talked about in so much detail today, that is getting stabilized because one hydrogen bond, another hydrogen bond, another hydrogen bond, one on top of the other, what they are doing is they are forming pi-pi stacking interactions among these sugar bases. Again, this is some chemistry that you need to go back to yourself. You can just Google it if you don't want to go back into too much detail. But these pi-pi stacking interactions provide an immense amount of stability to the DNA structure as a whole. But the backbone is what is made up of phosphodiester bonds only. In fact, it is called the phosphodiester backbone for this very reason. So maybe you can just revise it up for your own good. But yeah, there's a distinction between these two bonds. Yeah.
One confusion is still there. DNA forms, they are different. But one of the DNA form mentioned, I think A or B, the groove is at a 10.5, means one loop that looping is occurring at 10.5, 10.5. Does it mean one loop is occurring at 10 and another loop is occurring at 11? Because in next
No.
it has 11 and in third looping it is at 12. So it's a fine key nucleotide 1 to 1. They are forming a loop. So they are either 10 nucleotide or 11 nucleotide.
No. So what happens there is it got disconnected in between. Right. So it can never be half a base. What it basically means is that when DNA structure is studied, sorry. Yeah, sorry. Something is wrong with my internet. But yeah, basically what I'm saying is when the DNA structure is studied, what it basically does is that a region of DNA studied. Okay, say suppose you are studying a stretch of DNA. So say for example, you have some DNA where there are 20 bases per turn. Okay, and you are studying 40 bases and you are seeing that there are two turns in it. What will you see? You will see there are 40 nucleotides and there are two turns of DNA. You will simply divide 40 divided by 2 and say that in each turn there are 20 bases of DNA. That is how it is studied. So in that way, they averaged it out and they understood that there are approximately 10.5, which means that in one turn, one complete base is not there. At the turn, there is some part of the base which is there, or when you take two complete turns, you are getting 10.5 into 2, which means you're getting 21 bases in 2 turns. That is how they studied it out. It is not that there is half a base or anything of that sort. That is not feasible. Compare that to the A form of DNA. There you see that there are complete 11 bases per turn. So this is how studies in structural biology are simply carried out. Similarly, if you look at the way DNA is wound around histones, the way I showed with that mouse and everything, even there are, I think, 10.5 base pairs per 10.5 turns that are carried out for each wrapping that is around a histone. It's never that half a turn can happen. It is just that so many bases are accommodated in so much area. So it is just simple maths which is divided. Just take that example of 40 bases observed in 2 turns, so you average it out to 20. If there were 30 bases that you saw in 2 turns, it would be 15. So it is something like that.
105 bases in 10 turns. I thought they measured.
In one turn. No. 10.5 bases in one turn. One turn of DNA. This one turn.
5 in one turn means at least 105 in 10 turns if they had measured. So there must be one irregular. That's what I'm asking. So DNA
right. You do not get a complete base in one turn, maybe. Yeah, you can think of it that way also, that you study 10 turns and you get 105 bases from there. Same. The analogy remains the same.
It means 20. They had counted 20 turns, then it becomes 210 turns. That's why they had made it 10.5.
Right, right, right. Okay, so I had a few questions for the next class as well, but
Sorry,
we
madam.
can discuss. No, no, no worries. I can discuss it in the next class because I think we are done for today. But basically, we have covered most of the topics that were to be done for the first week. So it is around the central dogma, the
Thanks.
structure of DNA.
Nicely explained. Thanks.
Yeah, no worries. The structure of DNA and the overall process of replication. In the next class, we'll go into transcription and translation as well. And we had a few more questions prepared. I will upload the slides so you can go ahead and check it out. Those are just preliminary questions which might help you with your assignment. The answers are also marked there. So that is pretty much it. And if you have any doubts, we don't have more time for today, but you can certainly keep them noted and we can pick them up in the next class, or you can even go for the other discussion sessions you have tomorrow and I think day after tomorrow. So thank you for joining. I'll stop the recording now so you guys can leave. Good evening and bye.